First of all, what is a pathogen?
A pathogen is any disease-causing agent that can be living or non-living. Common pathogens such as bacteria are unicellular pathogens with a cell membrane, DNA, and organelles that undertake cellular processes making it living. If certain harmful bacteria enter an organism, they can multiply, causing infection and dangerous to the host.
Despite being obligate pathogens, bacteria can be transmitted to an organism through inhalation, ingestion, or enter the body through an open wound.
However, there are many both physical and chemical defences within the body that can help prevent the invasion of bacteria. For example, a thin layer of mucus lines the respiratory system, trapping pathogens physically whilst destroying them chemically through the release of certain enzymes.
The thick layer of skin that surrounds the more vulnerable innards of humans is considered to be the first line of defense against pathogens. If it were to be breached by, say, a thorn, bacteria on the thorn would be present, triggering what is known as the innate immune response, the second line of defense.
Therefore, the immune system must be able to recognise its own cells from foreign ones. Antigens are any substance that evokes an immune response, and most cells in the body are distinguished as being self-antigens due to the MHC1 markers on their surface. However, the foreign bacteria would be surrounded by antigens that would be recognised by receptors as being non-self, initiating an immune response on that antigen.
Cytokines or signaling molecules are released, causing mast cells to release large amounts of histamines, initiating an inflammation response. This entails the dilation of the blood vessels, allowing more blood to flow, as well as increased permeability, allowing cells known as phagocytes to reach the site of infection.
Phagocytes are mobile white blood cells or leukocytes that ingest microbes. These include the smaller yet more common neutrophils, and the less abundant yet far more efficient phagocytes include macrophages and dendritic cells.
Macrophages and dendritic cells are known as antigen-presenting cells. Unlike the rest of the body cells, these phagocytes contain MHC2 markers, which bind to the remnants of the digested microbe and present them externally as antigens.
Phagocytes engulf pathogens in the process of phagocytosis. The engulfed bacteria forms a phagosome within the macrophage where it fuses with the lysosome, now digested and expelled from the cell. However, as this is an antigen-presenting cell, the antigens remain on the outside of the cell.
The lymphatic system is a series of vessels that carry lymph fluid leaked from the blood into tissue, around the body, towards the heart, and towards the secondary lymphoid tissue, otherwise known as lymph nodes, the site of the adaptive immune response.
The macrophage will travel through the lymphatic system towards the lymph node to communicate with the adaptive immune system, the third line of defense.
Lymphocytes are leukocytes, derived from lymphoid stem cells. T lymphocytes mature in the thymus and differentiate into cytotoxic T cells and helper T cells.
If the antigen presented on the outside of a macrophage corresponds with a receptor of a helper T cell, that cell is alerted and begins replicating more cells with that specific receptor, clonal selection, releasing cytokines, chemical messengers, into its surroundings, activating cytotoxic T cells. Cytotoxic T cells then detect specific infected cells, secreting death ligands and triggering apoptosis of cells infected by the pathogen.
It is important to understand the difference between necrosis and apoptosis. Apoptosis is the programmed death of a cell, whilst necrosis is the inevitable destruction of a cell due to subjection to extreme conditions, such as disease or extreme temperature. Also triggered by the natural killer cells with the innate immune response, apoptosis occurs when a death ligand, or signaling molecule, attaches to a receptor of an infected cell, extrinsic or intrinsic, initiating a signal cascade, causing the mitochondria to release caspases, killing the infected cell. This process is the cell-mediated immune response, the third line of defense.
In the humoral immune response, B lymphocytes are large cells that mature in the bone marrow coated in membrane-bound antibodies that act as receptors. When the naive B lymphocyte comes into contact with a corresponding antigen, it is activated, cloning itself. The lymphocyte differentiates into memory cells which retain the genetic code for that antibody, whilst plasma cells act as antibody factories, filled with endoplasmic reticulum, producing free antibodies at a rate of 2,000 per second.
Antibodies are immune system-related proteins called immunoglobulins. Each antibody consists of 4 polypeptide chains, 2 heavy chains and 2 light chains, joined together to form a Y-shaped molecule, as well as a variable region, which forms the antigen binding site.
The antibodies released bind to the antigens of the pathogen, immobilizing it in a process of agglutination, preventing further infecting and allowing phagocytosis to occur more easily.
This also activates the complement system, in which complement proteins opsonize or surround the pathogen, immobilizing it, whilst attracting more phagocytes and opening a membrane attack complex, or MAC, breaking down the cell membrane and interrupting the life cycle of the bacteria, therefore destroying the pathogen that remains at the initial site of infection.